Coming up next, Dr. Marc Gerstein from Yale University explains the essence of life through network theory.
Netcasts you love.
From people you trust.
This is Twit. Bandwidth for Futures in Biotech is provided by Cachefly at cachefly.com.
This is Futures in Biotech, episode 83. Bioinformatics, essential gene names skewed in a network of blame.
I believe that biotech is the next frontier.
Probably the greatest intellectual revolution that's ever taken place in man's history. DNA is the code for life. We're actually beginning to understand how life works, which I think is something that's mind-blowing in and of itself.
There was going to be a genetic component to aging.
How long was there the extension?
About 30-40% for humans. That would equate to something like 20 to 30 years.
How close are we to actually having a therapy of something?
Ballpark, 10 years.
It's potentially one of the things that will end up rocking the world the same way that people said, oh, the sun's the center of the universe, oh, this and that and everything. And now here's somebody who can come out and say, hey, look, here's how we compare it to our closest evolutionary relative.
Welcome to Futures in Biotech. I'm Marc Pelletier. Today, we have Dr. Marc Gerstein, who's the Albert L. Williams Professor of Biomedical Informatics, Molecular Biophysics and Biochemistry, and Computer Science at Yale University. He's one of the top he is the top bioinformatician in the world, in my opinion, my humble opinion. I'm putting him on a pedestal. He's been a guest, a host, co-host, and a panelist and subject matter expert on Futures in Biotech, and I'd like to welcome him back. Welcome to the show, Marc.
Thank you.
And this time we have video. Hey, so I was a guest last night on Dr. Kiki's Science Hour, and I was for the first time on the other side. And one of the things I realized is it's always nerve-wracking at the start. So before we get into the hardcore science, which it will be, how about can we ask you a little bit about how you became a professor in 3 different departments, and which seems very, very cross field, or it's a very, very wide spread of areas of expertise. How did you get into science, and how did that lead you to being a professor at Yale in 3 different departments? That's something that's it's quite inspiring.
Well, I've always been interested in science, and I think I started out being interested in science in probably kindergarten or even beyond. I just like playing with molecules, at Lego, building things and stuff like that. I loved the DNA molecule when I was little, the helices and all that stuff. I thought that was really fun. And then when computers came out, and they weren't out when I was very, very little, I have to say, but when they came out, I really liked playing with computers. I think I got one of the early Apple computers, and I just enjoyed playing with it all the time. And
what was your first machine?
Oh, actually, I think I got something even before an Apple. I think I got one of those Commodore things. And
Commodore 20?
I think it was a Commodore 64.
Cool.
And I got that, and then I got an Apple, and I really like an Apple, and it's amazing. I'm using a Macintosh now. I'm very happy about that. All those years, we had a brief period of using the PC, and I liked computers, and I liked computing, and I liked molecules, and it's sort of natural that I would end up kind of doing computations on molecules, and so that's what I kind of ended up doing. When I was in college, I was a physics major, and they didn't really have computational biology or anything like that. And, I kind of got interested in biophysics and computational biophysics, and when I went to grad school, I tried to put those things together. They really didn't still have a proper kind of computational biology thing, but as times went on, after I've started at Yale and now, more and more they have proper programs in this field where they really bring together the computation and the biological sciences, and that's what really my appointment at Yale is supposed to do. I mean, I'm in molecular biophysics and biochemistry, and also in computer science, and my my title is professor of bioinformatics or biomedical informatics, and my real focus there is computational biology.
Wow. So you had when you started as a kid, did you ever make a DNA helix with Lego?
Not with Lego. That's a very good question, because that's actually very hard to do if you think about it. It's very hard to do. I've tried doing that. You can make helices, but if you think about it, it's actually very hard with Lego. But we had a little science project. I remember when I was in I think it was like in 3rd grade or 2nd grade. Just making things with like metal and everything. And I was really into making the double helix, and I still have that model. It's
It's cool.
an accurate representation, but it really consumed a lot of my time when I was little.
You walk into the old biophysics departments, right? You see the protein models where somebody actually did use cork and toothpicks to once the X-ray structure was determined, that's how people would have to visualize a protein structure. There's no way to do it on a computer. If you're in 1978, you're really not going to look at Star Wars, right? The way they had the high-res graphics of a interplanetary capable species of humans, right? They had computers that just had little vectors. So if you wanted to build a model, you did all your calculations on hand and by paper and by maybe poking the little piece of paper and running it through one of those Steve Gibson-esque computers. And then then it would give you the numbers, and you'd have to sit there and build it with corks and toothpicks. Didn't James Watson just do it with cutouts? Right?
Oh yeah, I mean all the original molecules people built obviously were, physical models, and there was this transition that happened. I think probably sort of after I really got into it, where everything moved into computer graphics and looking at it on the computer. But what's interesting now is we have the advent of course, the computation, but we also have 3D printers. So now you even have the extreme of looking at the molecule on the computer and actually being able to print it out in a three-dimensional representation. So I think people will go back to actually playing with physical molecular models soon.
Right. Well, I'm thinking, if HP, you think they charge a lot for their 2D ink.
Yes, no, I'm sure. Yeah.
Oh my God, yeah, God, they'll give you the printer for free, of course, a 3D printer, but it's going to cost you like $500 for the ink. I guess it's kind of like a plastic polymer, right? That just they melt on layers and.
That's right.
Yeah, I've used some equipment in the lab that was printed on 3D printers. Some valve systems for some impedance-based cell microfluidics devices. So let's talk a little bit about networks. You do a lot of work trying to. I guess one of the things that characterizes your work is that you try to get a comprehensive look at extremely complex biology. And, I have a very reductionist approach to my science, which is one protein, one drug. And that's what I can handle. And so I'd really appreciate if you could explain a little bit about your work and how you can sort of piece it together and explain it.
Sure. I mean, networks, I think they're very appealing to a lot of scientists because they do give you that kind of global or systems view of the whole cell or maybe a whole organism even. And at the same time, one of the really nice things about networks, I'll explain in a bit, is that they work in many different disciplines. So you can sort of see the same idea in biological sciences and kind of map it onto more commonplace contexts. And I think personally that's extremely important because people have very little intuition for molecules, particularly biological molecules, and I'll explain that a little bit in my talk, but they really can't think that easily about them. They can think very easily about social situations or more commonplace objects, and I think if you can make a mapping between them, it really helps you understand things. And that's one of the big appeals of networks.
I have to admit, in my 3rd year undergraduate, I read a genetic engineering text, and then it kind of hit my brain, and yes, I could visualize DNA, and I could really get gene structure, and all of a sudden it made sense. But still, one gene at a time. I'm not a systems analyst. I can't do it. It's just so do you have the slides, Bert? Do you have the slides? How do you go?
Well, that's fine. That's good for a first one there. Yeah, that's the second slide actually. That's good.
Okay, we're looking at a slide that has a chromosome stretched out, some annotation to that chromosome, and it's chromosome 7. Just for the listening audience, and and wow, there's some like chart. What's that? I'll let you explain it.
Well, yeah. So this slide is just supposed to represent kind of one of the problems people grapple with that they use networks for is people want to understand the genome. That's really the fundamental challenge in a lot of biological science now. What does the genome mean? One of the ways of thinking about the genome is this sort of list of molecules, and this is a picture of that list where you just have a linear chromosome and you have kind of little arrows to where all the genes are. And the question is, how do you think about the function of all those molecules and so forth? And and that's kind of the overarching problem that I think people address with networks. And actually, if you go to the next slide, that's the one with the little 3 on it. That one shows a little bit about kind of the traditional way people in biological sciences think about the function of a gene. And I don't have to go into the details of it, but the point is what...
Yeah, that's me. I get that.
yeah. But the thing is, there is a tradition in biological sciences of, I have a molecule, how do I describe its function? And, often what people want to do is that they want to use words. So they want to give the molecule a name, and they want to sort of describe its function by a sentence. So this is a well-known molecule here, and there's a little sentence that describes this molecule. It's called elongation factor 2 and how it's involved in translocation. This is the kind of traditional way people describe function. But then the question you have to ask, of course, is, if you go to the next slide, it sort of shows that you have to think a little bit about how this single molecule view is going to scale to tens of thousands, maybe even hundreds of thousands of different molecules that are involved in a cell. And you might say, well, I'll just have 10,000 sentences or 10,000 phrases to describe all the things. But there's a lot of problems with doing that systematically. I mean, first of all, it's actually hard to correctly use language to really describe all the functions of a molecule. And molecules have many different functions. Sometimes one function is carried out by a number of molecules. Other times you mean different things by functions. There's the role in the cell, there's the type of biochemistry the molecule carries out and so forth. And there's a lot of tricky things. And one of the other issues too is when you really drill into biological science in detail, you see that they've constructed this very complex language and names and stuff like that. And some of this stuff I think is just funny and fun. And I have a little bit of an aside here, which I thought you might find interesting about this type of language. And so on slide 5, there's a picture of there's a picture of a very famous Vincent van Gogh picture called Starry Night. And it turns out, a while ago I just stumbled on this gene name called Starry Night, and I was reading about it. So you might say, well, how does something get a name like Starry Night, and how does the word Starry Night actually describe the function of something or what it does? It's kind of very convoluted, but it's kind of interesting. It turns out that the person who named this gene, they noted that it was mutated. It changed the bristles on the eye of the fly to have that pattern that's shown in the bottom right. And the person was very erudite, the person who named this gene, and they said, geez, this looks a lot like this very famous painting by Vincent van Gogh, which is Starry Night. And so that's how they named it. And when you understand this whole little story, it's, that's really cool. You actually, that name Starry Night means something. But then you have to say to yourself,
amazing.
It does this.
By the way, we have a lag, and I'm looking at the picture here of the Starry Night painting, and then the bristles on the eye of the Drosophila, and it's scary how life imitates art here.
Sorry, it's scary. Scary what?
It's scary how life imitates art.
Oh yes, well, or maybe
Brilliant.
art imitates life. But
It-
yeah, no,
it
it is interesting, and I think that it's actually a great name. The thing that's interesting, it's a great name for something when you understand the connections. But if you don't, it actually is completely perplexing, and you have to say to yourself, well, how does this type of idea scale to hundreds of thousands or or so molecules? And it doesn't. I don't think very well. And that's kind of the problem. And actually on the next slide, this is just kind of fun. I like to, this is kind of a sort of talk I tend to give a lot. And I actually kind of got really into this naming these bizarre names in biology. And I actually created an ontology, kind of classification of naming pathologies. And there's just some really fun names that you can actually look at the different types of names that people create. And so, for instance, there's two genes. One's named Kryptonite, and the other's named Superman. And it's actually interesting. What's happening there is they're transferring a naming system from someplace else. It's obviously the Kryptonite and Superman from the comics to the molecules. And if you understand the comic story, you'll understand sort of what the molecules do. And there's other times where people make really interesting allusions in molecule names. Like, for instance, there's a molecule called Uri, and you might say, well, what's that mean? It's named for Yuri Gagarin, one of the first cosmonauts. And, of course, this molecule has to kind of do with sort of balance and, sort of falling down and so forth. And, there's a lot of these interesting things. And I actually
Is there a
find
tribbles protein?
Yeah, there's tribbles, and it has to do with reproduction. You can probably guess. Well, no,
Reproductive gene named tribbles. That's great.
Really,
or Blue
really
X, right?
fun. But again, it's one of these things where if you drill into each of them, it's a lot of fun. That's why I thought I'd kind of show this. But again, it's not a type of thing that's going to work for 10,000 things in the genome.
Drop Dead. I like that one.
Drop Dead is interesting. That's humorous, but it describes what it does. If you have this this particular gene is associated with a mutation such that the organism drops dead really quickly.
That's nuts. So I guess, well, yeah, but the way it worked, right? I remember back in the day, we'd do some kind of biological study looking for proteins and searching out functional interactions of proteins with other proteins, and you'd find a new protein, you'd run the DNA sequence, you'd come back on Monday after exposing your film for the weekend, and it was very, very, very exciting, and you wondered, did I get the whole open reading frame, and you're using radioactivity, and the lab was a mess, and it was a little dangerous, and so you'd look at your A's, your T's, your C's, and your G's, and you do it by hand, and then, it was so much work. Giving it a name like tribbles is makes it really fun, makes the
Well, yeah, no, actually, there's one name there called Yippee, and supposedly the gene was named because the person just felt like Yippee when they discovered this particular gene.
There's the sumo, right? There's all kinds of
sumo too. Yeah.
I guess it has. It was these. It's kind of like in the movie with Cusack, there where he's naming his albums, or he's positioning his albums by chronological order, by biography. All these genes were named over time by individuals who had some tie to the discovery, and they were done very, very slowly. But I guess in today's day, when you're doing a genome, high fidelity, yes, thanks, Beatmaster. When you're doing an entire genome in about 3 hours, I suppose you can't do much more with it, right? You can't. It's hard to organize and visualize.
Well, that's the issue. I mean, this type of stuff, it's really interesting, and in a certain sense, you might say very baroque in a way, but it just doesn't work well with the genome in a few hours, or the very large scale biology. And you can kind of see it's kind of almost like biology is pushing up to the boundaries of language, really, to really be able to describe all these things well and so forth. And English language is not always the right sort of tool for this type of thing. So in any case, what you got there is that's an ontology. Actually, on the next slide, on the 7 one there, I thought this would be kind of fun. This is you can actually plot for each gene name how often you see that in the biological science literature versus how often you see it on the web. You know, this was done a while ago, so we used Yahoo as opposed to Google, and it's really neat because there's some where they're kind of proportional. You see the name more in the biological science literature, you see it more on the web, but there's some where you don't see it very much in the biological science literature, but it's really common on the web, and you can see what's going on there. These are genes like cat or dog, or where you have the layering on of a kind of common usage onto the gene name, and it's kind of neat. You can see that there's a certain fractioning of these names that just have a life onto their own, in the sort of popular world, beyond the biological world.
What's there's one gene that seems to be the highest hit on Yahoo and highest hit on PubMed. Do you know what gene 1 and 2 are here?
I don't. I should know. I think they have little numbers because in the paper, we probably called them out, but I don't remember off the top of my head. But that is actually kind of interesting what those are.
Well, there are some. Well, knowing what the nature of the internet is, I wonder if these are reproductive.
Yes, no, that's.
Sorry to bring you down to that level, but these probably have some name that are relevant to reproduction.
Yes, no, that's actually it's a lot of stuff is fun. I mean, you're probably right that a lot of the names that are really popular, really skewed. They fall in certain categories for sure.
I won't say anymore. Okay. So I guess Google would, the internet is the internet. So by comparing the PubMed hits to Yahoo hits, you're inferring that language has
well, you're seeing the distortion. That you're seeing the way a lot of gene names are kind of overlaid with popular terms, and like I said, you can kind of just sort of see that for a lot of gene names, they're kind of layered on with a lot of things, and again, I think you can kind of see the Baroque kind of nomenclature things that we're seeing a lot in biology, and I think this is a good thing. I mean, it's fun. It's frankly a lot of fun. But again, it's not the thing that scales. And actually, if you go on to the next slide, I'll just kind of walk you through this. And so people have been thinking about what to do. How do you be more systematic? And they have, there's a whole world of systematists who, they have these what they call ontologies, or controlled vocabularies. And you can kind of get a sense from looking at these little figures. They kind of have categories, and things fit into bigger categories. And there's different ways of doing this. And that's one approach. There's a very popular biological system called the GO system, for a gene ontology system, that kind of classes genes this way. It's a very useful system. But then in the next slide, on slide 9, I kind of give you the network perspective, and the network perspective basically is you don't so much think about what something is, but you think about what it's connected to, or its context in the network. So it's like, it's not who you are, but it's who your friends are. It's like you're defined by your connections. And if you think about it for a second, I in a certain sense, even in kind of social context, that sort of makes sense a little bit. It's a way to think about people. And actually, I think for molecules, a lot of times, their context is the most important thing about them, and the other molecules they're around. And so the picture just shows, different representations of networks. There's high throughput representations. There's more traditional representations. But the key idea is that central molecule notch, which is again one of these, names that is obviously an overloaded English term. It's actually not being defined by that word notch, but it's simply being defined by its position in this overall network. And then I got a few more slides that just give people a little bit of a sense of the why networks are kind of so popular now. And the next one, which is slide 10, just gives you a bit of a sense that what biologists, or I'd say scientists, really want is, they want to, of course, go from that 1D parts list of the genome that we started out with, and they really want, detailed biophysical understanding of how everything works, predictive understanding, how each of these things work as machines, almost to a level that you don't really even need to talk about them. You can predict how they work almost, like I said, as machines. But the truth is that's a long way away for, the 100,000 so molecules in the genome. And so the network, or the kind of wiring diagram representation, is this kind of very convenient middle place that gives you some sense of what's, who's talking to who, who's connected to who, and so forth, and gives you a little bit more annotation or information than just the parts list from the genome. But at the same time, it doesn't require as much knowledge as, let's say, the detailed, say 3D understanding of all the molecules. And I think that's one reason why their networks are really popular. Another.
By the way, before you go on, I want to just explain this to the listening audience here. The slide that you're showing here it's got three basic panels. The first panel on the left is a circle map with little hash lines going through it, and it represents the genome of probably a bacteria, right? One chromosome?
That's correct. Actually, that's one of the first genome sequence, I think. I think it's Haemophilus influenza, actually.
And if you look at that, you get absolutely just the parts list, because it doesn't give you any more information. But you just got their position within the chromosome. Then you go to the one that looks like if those in Quebec, a pom-pom that you might find on a hat on a chick.
People use from hairball. Hairball is the kind of
hairball, which it looks exactly like a hairball, or a sparkler with lines going out all over the place. And so each individual node of that represents the product of a gene, or a gene that has interplay with multiple connections. So you see this, like, it's kind of a mix between a hairball and a sparkler. Then the third one is the actual three-dimensional structure of the gene product, which is the protein. So networks occupy a midway point in the terms of level of understanding. Right. So when we were cloning genes, back in the heyday of the 90s, where you would look for mutation in yeast that caused a biological function to be changed, you isolate the DNA and you compare it by its sequence. You knew the sequence, you could read it, so you're on 1D. But then by sequence homology, you could predict its structure a little bit, and that could predict a little bit of what it was doing or how it was doing. But then if you did something like a 2 hybrid screen where you're looking for the proteins that interact with it, you could fit it within a functional pathway and then nail it down. And that's your second figure here, which shows how just having the network information can give you a tremendous amount of information as to how that gene is working. And by the way, that could be, you're looking for the proteins that modulate the function of a chloride channel, which might result in a disease like cystic fibrosis, right? So while this seems theoretical, when you're actually trying to tackle a major problem, you need to know, and the quicker you know what that information is, the quicker you are towards a cure.
Sure. I mean, it's often the case that the diseases and sort of anomalies really result from many many genes working together in these kind of systems. And like I said, often the way people represent the many genes working together is in the framework of a network or pathway or some multi-molecule representation. And I think that's one of the reasons these things are so popular in biology now. Now, another thing that's interesting is if you go the next slide 11 there, I think one of the really powerful things about networks is the point I was making earlier, is that people in biological science are very interested in them and think they're great, but there's a lot of other disciplines that are using networks now. So there's obviously neural networks, there's the internet, computer networks. People look at social networks and so forth. And in the center, I have this book by this very famous network theorist named Barabasi, who's talked a lot about networks properties in general. And one of the neat things is this community that's being developed now that talks about network theory or networks kind of as an abstract concept. And the language they use crosses all these disciplines. And so it's a way actually for people to communicate biological ideas in actually much easier to understand language than the language of Starry Night or the complicated gene names. And so I think one of the most powerful things that networks provide to biology now is a way of communicating the complexity that people in other disciplines can understand. And also, like I said, providing analogies, as I'll just describe in a bit, where you can think about a biological science system as a social network or as, more in the way you think about a computer. And this a lot of times helps you in thinking about things. And so I think this is one of the nicest things about networks. And actually, the next slide is, I think, a good illustration of what I mean by this. So if you go to slide 12, this is just a picture I took out of the New York Times, or two pictures I took out of the New York Times. I think you can kind of get a lot of this puts together a lot of things. So I've been talking about how does the network describe function, and how do we use analogies to really understand molecular functions. And so the top right-hand thing, that shows a picture that they call the blame game. And what it shows is a bunch of arrows with heads of politicians, and the arrows kind of show blame being ascribed. And actually, if you look at this, you probably recognize all the people on the cartoon, but you can almost kind of see, like, what's kind of going on, who's in charge, frankly, and where all the kind of blame is going. And I think it gives you a sense of the function of these people, even if you didn't know who they were. And likewise,
let me just describe it a little bit for a sec here. You've got Rush Limbaugh blaming Mayor Nagin, and he is blaming Michael Brown from FEMA, and George Bush is blaming Michael Brown from FEMA, and he's blaming the churches, and Bill Clinton is blaming George Bush, and Bill Maher is blaming George Bush, and Governor Kathleen Blanco is being blamed by Rush Limbaugh and by the news media. This is fantastic. Okay.
Well, it is. But the thing that's neat about, like I said, is you look at this kind of network of blame, and you kind of get the sense of, the phrase "where the buck stops here." Well, it does in a sense. It stops. You can see up there at George W. or something like that, and he's you can see he's got a self-blame little loop.
Yeah, he blames himself.
Yes, no, but I think it gives you a sense of his position.
He's a leader. Yeah.
And again, people have an immediate intuition for that, what's kind of going on, because it's a social situation that they're accustomed to. Now, if you see the same picture for molecules, they won't have the intuition, but if you can map it, map the molecular picture to the social picture, I think it helps people a bit understand things. And likewise, the one on the bottom left, this one's called guilt by association. It's another just clipping from the New York Times. And you can see, I think it's kind of obvious what's going on here. You've got this, I can't pronounce this guy's name, but Rod B., who's this governor who got himself
Blagojevich.
Yes, yes. And you can see, what does guilt by association mean? You can see the path of connections in a sense. And again, people use that term guilt by association when they're talking about molecular function, and here you get a much more intuitive idea of it.
Right. There's Anton Tony Rezko, who's connected to William Cellini. Well, I don't want to mention names.
Well, these people are very, very public characters. I mean,
John Harris, chief of staff, and so if is red guilty and blue not guilty?
I think that's my interpretation of this figure. Yes.
Right. So if you know someone, you're guilty. Well, that does reflect biological function. If I'm looking at the first one that you described here, the blame game for Rush Limbaugh only has arrows blaming away from him, and I can imagine a lot of proteins that have that particular activity. It might be a protein that modifies, for example, adds methyl groups to DNA. It doesn't receive methyl groups. It just gives methyl groups. and some proteins like kinases that are way up high on the signaling pathways, which act sort of like dip switches to major biological function, they might be phosphorylating themselves, and that's kind of the leader position and high up on the rank of proteins to watch, particularly for disease, because if they're mutated, they disrupt a lot of systems. I suppose people die in those situations, but
sure. I mean, what people are very interested in the framework of biological networks is they want to kind of find these regulatory cascades or kind of chains of causation, that are potentially leading to cancer or even just leading to a normal biological function, and they of course want to modify these cascades, or at least kind of understand how biological regulation takes place. And I think to some degree, I mean, it's obviously not the same as in a social situation, exactly the same, but I do think some of these analogies are quite useful.
I'd like to take just a quick second. We don't have a sponsor, but I'd like to do a little bit of station ID. You're listening to Futures in Biotech on Twit TV. And today we have Dr. Marc Gerstein from Yale University explaining the sort of the how to develop a major nomenclature for a complex system. That is life, I suppose. I guess that's really what you're doing, right? You're building up to establishing a framework. Well, I don't want to. I'll let you continue to explain, the inner workings of the molecules and the chemistry of life, which hasn't been done yet per se. But I'll let you continue. So
that is definitely the idea. I mean, I wouldn't say obviously just myself doing, but I do think that the idea of all the people in this kind of systems biology and kind of network community is they do see this as a kind of framework to understand the cell and to understand how the genome's functioning. I mean, that's really the thrust behind it. The whole discipline. And I have a few more beginning slides. I'll just quickly say, I don't know if we have to belabor them. The big thing also that's important in biological science is that you probably just jump right to slide 14, just 2 down. Is that a lot of the types of connections that people study in biology can be represented as different types of networks. And there's the proteins are interacting and touching. That makes one network. There's the metabolic pathway of the sort of small molecule being passed down between different enzymes. That makes another network. There's the regulatory network of the transcription factor regulating its target. And again, that makes a different network. And this picture just kind of shows...
That's kind of like a Google Circles thing.
Oh, yes. Well, yes, Google Circles is maybe a little less regulatory, but it's not.
It's true.
It looks like that. Well, I think that, like I said, I think that's the key point, that these pictures do have immediate intuition for people. And that is different from a lot of the other situations you have in biology where people don't have that quick intuition. And so if you go to the next slide, I don't know if we're going to really have time to go through all these particular things, but I did have a little bit of a layout of different things that you can kind of talk about related to networks. And so there's a certain background of terms that people have, that people think about related to networks. And then I thought that what might be good to do is to talk about some of these types of network comparisons, which I think they're very easy to, like I said, easy to grasp. And two that we've been very keen on are comparing the regulatory network to kind of a government hierarchy or social hierarchy, and then comparing the kind of regulatory network to the way a computer operating system works. And then the final thing is, which I don't think we'll have probably time for, is to look at how networks change in different environments, and that's kind of an interesting thing people talk a lot about too. I don't know, I mean, how do you want me to do this? Do you want me to just kind of quickly jump through some things, or spend a lot of time on one thing, or how do you want it to go?
Well, I don't... I'm just trying to follow.
Why don't I just plunge in? I'll plunge in. I'll give a little bit of background, and then I'll jump into the comparisons, which I think are actually the most interesting stuff, and I'll focus on the sort of, how should one say, immediately obvious types of things. So one of the things, okay, so if you flip there to slide 14, or sorry, slide 17, one of the immediate things that people look at in networks, a very simple idea, just that that people talk about the degree, sort of like how many friends do you have? If you're the blue node, do you have 4 friends, 5 friends, whatever? And then they talk about path length. How many, if you're one blue node and there's another blue node, how many links connect you? Very simple ideas. But those simple ideas, there's some neat things that people have found. And so if you go to the next slide 18, one of the things that people have found looking at networks is this concept of hubs. And so when they plot how many kind of friends or neighbors a given node has, they'll often find in many networks that there's a most nodes, or sort of center points or molecules, whatever, have only a few things they're touching, and then there's a few things that have a lot of things. And they sometimes use the term a power law distribution or heavy tail distribution to describe this type of relationship, that there's a few things that have a lot of connections. And if you think to yourself, what are those few things that have a lot of connections? They're hubs. And this is a commonsensical word that people use. And you can think to yourself how these hubs, they act to kind of structure the network. And so the picture to the right-hand side shows a little network, and it shows the hubs with a little bit of color on them. Sort of obvious, you can see those hubs are very important, and people have an immediate grasp for this. So that's one sort of term that people often use. And now if you go to the next slide 19, one of the things people did initially in kind of network biology is they said, well, geez, hubs appear to be very important in just the abstract connectivity of a graph. Maybe we can actually show they're more important in terms of an organism. So what they did is, and this is the neat thing about biology that really separates it from many other disciplines, people can do an experiment. They can actually do a systematic experiment to test this. So people did experiments where they knocked out every single gene in an organism, and then they looked at the connectivity pattern of the genes they knocked out, and for each gene that's knocked out, sometimes the organism dies, and sometimes it lives. If the organism dies, the gene's said to be essential, and if it lives, it's said to be not essential. And what people found, which is kind of sort of fits with your intuition, is that that hubs, or things that are more connected, tended to be more essential than not. And this was early work that people did years ago, and there's been a lot of follow-on ideas, but this is one of the things a lot of people in network biology have looked at, this kind of idea that the connectivity is related somehow to important properties of the molecule. So you can kind of say, well, we don't know what this gene does, but it's a hub gene, and that really tells you about kind of where it sits in the whole molecule, and how important it is.
If you knock out a gene and it causes the animal or the organism, the yeast, for example, to die, then you know that you're not that, but you can expect that protein to be a hub protein, or for the most part, likeliness that it'll be a hub protein or a VIP for a very important protein.
No, that's correct. I mean, it's not a perfect correlation, but there's definitely a strong enrichment for hub proteins being essential genes. And like I said, it makes sense, and it's actually very satisfying to see that. So that's one sort of simple network idea. Now another idea that you can see if you jump to slide 21 is there's another concept of centrality, or another concept of connectivity in networks, which is a little harder to grasp, but is actually more important, it turns out, in regulation. It's this concept of what they call betweenness centrality, or bottlenecks. And it's a little more abstract. Imagine you have a network, and you kind of connect you look at the shortest path between every pair of nodes, or you have a social network and you ask, what's the shortest path between every pair of people? And then you ask, which person does all those shortest paths go through the most? and that person would be a bottleneck, have a very high betweenness centrality. And you can get a little, now that's a little abstract what I said, but if you go to the next slide, you can get some immediate intuition for this. So people kind of know what a bottleneck is. Think of the road network, and the next slide just shows a picture of the George Washington Bridge. That's a bottleneck on the road network, and it's because a lot of the shortest paths through any two different points all converge on that one link. So if you want to get anywhere in the Northeast, there's a very high chance that you're going to want to follow the shortest path. You're going to end up driving over the George Washington Bridge. And because of that, a lot of traffic, and it becomes a bottleneck. And people have immediate intuition for, if you're in traffic, what is a bottleneck? Now, again, when we're going to talk about molecular networks, we're going to use the same terms. And if you think, George Washington Bridge, when you hear bottleneck, you get a lot of intuition. And the next slide, slide 23. I hope I'm not going through.
No, this is good. So we've got just a quick review. We've got hubness, where you've got a lot of people grabbing your arms from so you're a 12, like you're an octopus with arms stretched out. You're the hub, and the lines connect to you. Right? It could be a subway station with 5 lines that merge somewhere in Boston, for example. Then you've got betweenness, which is a short route between 2 major systems. You might be the one that carries people over, and a good example of betweenness is George Washington Bridge, where you see a 12-lane, or it's more like 20-lane highway all merges because that's the shortest distance, but that leads to bottlenecks.
That's exactly right. And if you go to the next slide, it just actually shows just what you said. Where the neat thing is, you can, if you look at the little colors there, you can see a node or a gene, or like in a social network, a person could be a hub. And for instance, the orange guy and the green guy are both hubs, but some of those hubs could be bottlenecks. They don't have to be. And you could also have a bottleneck that's not a hub, and vice versa. And it just kind of gives you a little bit of a sense of that, what those types of things are. And then the next slide.
By the way, let me quickly interrupt and say that if you're trying to strategize a game like, what's it called, The Amazing Race, there's the bunching and the hubs, and basically if you have a bottleneck and it's a combination of bottleneck and hubs, you could probably strategize a way to get through the race and get around people quicker. So I urge people, anybody's interested in Amazing Race, they take a look at these slides.
That's an excellent point, because I think that like I said, the neat thing about these, the bottleneck and hub concept, they're actually very abstract concepts in graph analysis, and if they're presented in a very dry way, people don't get a clue what they are. But you, of course, confront them in everyday life, and you get immediate intuition. I want to do a quick journey someplace. How do I do it? And you get immediate intuition. Now, it turns out on the next slide, slide 24, this just shows that people have done some correlative work, and it turns out that in regulatory networks, the bottlenecks are actually more important, and they're more associated with the essential and important regulators than the hubs. This is just a graph that shows that particular type of correlation. And so this will come up in a second. You can kind of get some intuition why bottlenecks will be very important in molecular networks, because you can kind of see them as like there's information flowing constantly in the cell, and a bottleneck is a sort of choke point. There's just too much information wants to flow down through that spot, and that's why it's going to be associated with things that are very essential and so forth. And I'm going to come back to that in a little bit.
This could apply entirely to VLSI, right? Very large circuit integration.
Oh, definitely.
Trying to design a chip that's faster, right?
For computer networks, is a very, very clear, type of analogy, and people often, they'll talk all the times about bottlenecks and in the internet, that maybe are dealing with our Skype transmission right now. But that's a thing people think a lot about, and for sort of obvious reasons. Now, so just a little bit more background, just real quick. I'm going to just jump you quickly through all the way to slide 27. this slide shows if you can see it shows a picture of the human protein interaction network. And what it shows painted on it shows painted on it a little bit of an abstract concept, but it's the concept of which genes are changing a lot or which are varying. So we have, roughly 20,000 odd genes in the human genome, and some of the genes are varying more in the human population than others. And you might say, well, is there any pattern to that variation? And it's actually kind of neat. So if you paint the genes that vary a lot onto the network, you can see immediately, you'll see the picture, you look at it, you see, the genes that vary a lot, they tend to be on the periphery of the network. They're not in the center. Now, this is a picture, and actually in the next slide, which I don't even have to bother looking at, it shows more of the quantitation of it. But the important intuition is, think about it for a second. Why might you have the more varying genes on the periphery of the network? Well, you can kind of sort of see it's related to that kind of essentiality idea. Think about if you have a very complicated computer network, and you have like your central server that has all the files on, and then you have like some, client computers that are on the outside. You probably are going to play less with your central server because it's more fragile. If you change it, there's so many other things that are dependent on it. Whereas the things on the periphery of the network, you change and you mess them up or something like that. There's less things that are dependent on them. And so it's sort of obvious that... Say what?
You don't die. You got
well,
that's the color of your fingernail, and nobody cares. But if it's the mechanism for providing energy to a heart cell, then you're going to die, right?
No, that's exactly the idea. I mean, it's sort of like you look at a huge pipe network, and you said to yourself, geez, I'm going to throw a wrench at that pipe network, and where's going to cause the most damage? Obviously, the place where the pipes are all connected very tightly, if you mess up there, it's going to cause a big bit of damage, whereas stuff at the periphery won't mess up as much. And it's obvious intuition. And the really neat thing is that you see it in the reality of what happens in human proteins. The human proteins that are tightly connected, they're what the formal language is, people would say they're under greater constraint, and the proteins that aren't as connected, they can change more. They're under either less selection, or they're actually under what's called positive selection. So they're actually encouraged to change. And so that's some it's sort of obvious intuitions, but it's nice to see. So now what I'll do is I'm just going to skip all the way ahead to slide 30, and what I've done a little bit is I've given you a little background on why you want to look at networks, then some concepts in networks, like these information flow bottlenecks, and then a little bit about how variation. And so now I'm going to give you a little bit of intuition about what molecular networks look like and try to make some comparisons. Actually, go to the next slide. That's slide 30. So this is a social hierarchy. I made this slide a long time ago, and at the time, I guess George Bush was the president. And, he was at the top of the governmental org chart, and I'm sure you recognize all these things. And again, people have immediate intuition for what that is. So one of the things we developed a while ago, and shown on the next slide, is we took a molecular regulatory network and we tried to unravel it into a hierarchy. And so the next slide just shows a little computer algorithm that's trying to take a network and kind of figure out, well, what things are just regulated that sit on the bottom, and how to work up what things regulate those, and so forth. And there's a technical term for how this thing is searching. It's doing what's called a breadth first search. But the important idea is that you end up with a little hierarchy that has different levels. And then if you go to the next slide, that's slide 33, you actually can see the analogous molecular hierarchy to the social one that we start off. And so if you see there, you'll see this picture on the left of the regulatory network in yeast, that's Saccharomyces cerevisiae, and then on the right is a network in this bacteria, E. coli. And these are very simple organisms. And again, you can see right away there's some things at the top, they're sort of regulating and stuff like that, and then some things on the bottom. And so you get a little bit of intuition. Who's in charge of yeast? How's the government of yeast work? And you get a little bit of intuition from looking at a type of picture like this. Now,
there's a committee of 8. I see a committee of 8.
Well, no,
whereas
but
E. coli has 2.
The interesting thing, and I'll just show you in a second, you might think, now the quick intuition is that the things at the top are actually the things that are in charge, but actually what I'll show you in a second, it's not actually the case. It's actually the opposite way around, and it's actually kind of interesting. So if
it's democracy. It's a democracy for the people at the lowest end that are working their way up.
Well, that's actually exactly the right intuition. A lot of times, I'll show you this in a second. People on the top of these hierarchies are very important, and they're very influential. But actually, when you look at the runnings of like a country or a social organization, the person on the top can change, and the country doesn't stop. Things keep on going, and you see this. No, it's the same type of situation in a cell. So if you go now, just skip the next one, slide 34. This is a more technical thing on slide 34. But on slide 35, actually, it's exactly what we were talking about. So what you can do, if you go to slide 35, exactly what you were getting at, is we can take all the genes that are essential, and we can, and actually the essentiality comes in different flavors of like strength of essentiality, and we can map them onto that hierarchy, and that's shown by the little red dot coloring on this hierarchy. So you have this layer cake, and now I'm coloring each of the genes like a degree of red by how essential they are. And then you can say, well, where are the essential genes? And you might say, geez, the guys at the top, I think they're going to be essential, but it's actually not the case at all. It's the other way around. It's actually the guys in the middle and to some degree the bottom that tend to be the most essential. And that's kind of weird. That's very, at least immediately, it's a little different than what you might think. But what's also really neat, and I'll come to a rationalization of this in a second, that people can do other experiments where what they can do is they knock out each of the genes and they see that the organism still lives, but then they ask how many of the other genes are affected. And affected can take many different forms. It can be how many other genes are affected in their level of gene expression, or it can be how many other genes, or how much is the cell affected in how it grows. And then what you see is the opposite. You see that the things at the top, as you kind of expect, are kind of most important. If you knock them out, more things are affected. And so we call this kind of the paradox of influence versus essentiality. So it appears that in a biological system, the things at the top are influential. If you affect them, the whole cell is affected. But they're not essential, okay? And actually, I think this is kind of like sort of the president and the systems administrator. If the systems administrator in my lab disappears, my whole lab shuts down like tomorrow. It just ends. But if the president, when we had a transition from President Clinton to President Bush and then President Obama, certainly my little world it keeps going on. Now I'm of course influenced by that, and there's a tremendous influence these people have, but it's a kind of more muted influence over everyone. And I think it's a very nice sort of social analogy for what you actually see in a real biological network. And now, actually, the neat thing...
It definitely is a paradox, though, essential versus influential. I would have always thought that the essential were the influential.
Well, no,
I get your point.
It gets better. You can even reconcile the paradox a little bit. So if you go to the next slide, that's the
you stop the trash, and the system breaks down. But, you change the president. Yeah.
Well, I think
You're
it
right,
does
you're right.
reflect people's intuitions. Like I said, the very important leaders of the world, when they change, it affects people for sure. But it's not like a little community disappears. At least most of the time, that's not going to be the case. Whereas a lot of times there really are essential people that do specific jobs, you know, a doctor in a hospital or a person even, say, controlling a big power plant or something, whereas if they were suddenly to die or disappear, you know, it's just quite dramatic in terms of its effect. But in any case, this is this paradox. But the neat thing is you can actually reconcile this a bit in terms of the connectivity that you see in the network. So the slide 36, it shows that same picture of the hierarchy. But now what's plotted on the right-hand side is it's just sort of plotted what's technically called the out degree of each transcription factor, but what it means in simple-minded language is how many other things does that molecule regulate. Okay, so, you know, it's like if you're in an organization, people use the term, how many direct reports do you have? And so what's plotted on that graph is how many direct reports each, on average, each molecule in that layer has. And what's really interesting is that the molecules with the most direct reports are the ones in the middle. So the ones at the top, even though they sit at the top, they don't have that many direct reports. You know, the guy at the top, you know, he talks to a few people beneath him, but, you know, it doesn't like he has like 100 people reporting to him. And this is actually similar to what you have in a big organization in a company. You might have the CEO doesn't actually govern directly all the people. He has as direct reports maybe 5 people, okay? But it turns out in the middle of the hierarchy, that's where you tend to get regulators who have a tremendous number of direct reports. And the way to think about this is the factory foreman, you know, who has like literally 100 people who directly report to him. And what's kind of neat is we wanted to make this analogy a little bit more precise. So we wanted to actually find a real org chart and look at the number of direct reports. And so first we had the idea we'd grab like the US government org chart or the IBM org chart. And it turns out they're much more complicated than the yeast org chart. There are many, many more levels, much, much more complicated. You could never kind of put them side by side. It's interesting. You don't realize how complicated they are. So what we actually found to make a comparison...
That's hilarious, though, that, you know, evolution in time, a couple of billion years, can lead to a highly complex machine. And I'm talking complexity, not like like an analog watch with all moving parts. These are parts that make themselves and then disappear, and make themselves and disappear at different time cycles and different stages of life and different types of cells. I mean, think about the complexity of the human brain, starting from a single cell to the most complex machine in the world. Then you take its subparts, right? The actual. I'm not just talking about the cells, but then going on to the genes that regulate and make the products for the human brain. I guess maybe the human brain's more complicated than the U.S. government, but clearly yeast is a complex beast, and you're saying that the U.S. government is more complex with respect to hierarchy and
complicated. But what's neat is you might say what's analogous to yeast, and if you look at slide 37, we found this org chart that's kind of analogous to yeast, and it turns out this is the state of Macau. This small city-state has about the same level of complexity. And you can see it's got 4 levels there. But what's really neat is you get the same kind of overall thing, where the guy at the top, you know, you can see it's really clear here. He's got 5 direct reports. But the guys under him, they have more direct reports, right? Mm-hmm. And again, it matches your intuition about how you see social organizations. Now, the final thing to think about is, now, think about so we've introduced the idea that the things in the middle tend to be essential. We've introduced the idea that the things in the middle have more direct reports. And then there was this other thing that we talked about earlier that I'm going to bring in now, this idea of bottlenecks. So if you go to the next slide, what you can do is you can calculate in that hierarchy where the information flow bottlenecks are. You know, for each node, you can say how much like the George Washington Bridge is this node, right? And then you can calculate the average, what's called the average betweenness, or the average degree of bottleneckness in that hierarchy. And now, lo and behold, it turns out that it maximizes in the middle level. And the reason it maximizes is because those middle level guys have the most direct reports. It's sort of obvious that all the information has to flow through that factory foreman. And therefore, they become an information flow bottleneck. And therefore, it's actually quite reasonable that they're essential, if you knock them out. And so, see, this is the type of way I think the network connectivity actually kind of explains a little bit the phenotypically what you observe in terms of the molecules.
That's an incredible way to visualize life and the importance of each gene. And, God forbid, we were to create, so we can set up the framework and create a social network based on hierarchy. Can you imagine if Google Plus or Facebook could have hierarchy?
Well, no, actually, it's coming to actually. If you just skip all the way now to slide 40, it's the next.
You can monetize
that's
it.
Actually a point that you're raising. So if you skip to slide 40, now, okay, let's come back to a social situation. There are many types of hierarchies, and you might ask, well, maybe Facebook is maybe not the best example, but there's a lot of social situations you're often in that are hierarchical. Okay. And let's think about the different types of hierarchies that you might be in. The one on the far left is kind of the extreme hierarchy. It's the autocratic hierarchy. The military. Everyone's direct report up the command chain and so forth, right? Mm-hmm. And so the guy at the bottom reports to the guy above and so forth. And you can kind of see immediately how this type of hierarchy leads to information flow bottlenecks, because the guys in the middle, you can see how all the information is going to bottleneck right in those things. And military planners think about this, right? Because they think about, well, what happens to that little platoon, on the ground if the head of the platoon gets knocked out? If the head of the platoon is knocked out and all the information has to flow through him, what's going to happen to all the troops? It's actually quite a big deal. And so the kind of traditional autocratic hierarchy, it has a lot of issues. Now, if you look at the middle hierarchy, you have here what we and other people have called is a more democratic hierarchy, where you have a sense of some things are at the bottom, they're more regulated, and some things are at the top, they tend to regulate, and then there are things in the middle, but it's not absolute. And one thing can actually be regulated by a number of things. And one way I think about this is kind of like a law firm, where you might have an associate, quote, at the bottom or something like that, but he's reporting to a number of partners or something like that. There's not as clear sense of chain of command. And now what's neat is if you think about the autocratic hierarchy, and you think about that middle level, like that, the platoon leader, and you say to yourself, yeah, that's a problem. What if that person was knocked out? Then you imagine, if you go all the way over to the right, adding in like an extra link, like a kind of crossbar to the network. Now you make one of the privates at the bottom report to more than one platoon leader, right? And you can kind of immediately see intuitively how that's going to cut the information flow bottlenecks in that central level of the network, okay? Mm-hmm. And and you can kind of see how it's going to make it more robust, right? Because suddenly you don't have this issue. And so one of the things we did, and if you go quickly to the next slide, is we tried to see, do you see this kind of more democratic type of hierarchy in biological system, or a more autocratic hierarchy? And the rationale for the more democratic one would be it's more robust. And very quickly, actually, I'll just tell you, I don't even think we have to look at all these slides. Or you can look at that slide 44, but I'll just say it in words. What we basically found was that you can define a measure of kind of the more democratic or more autocratic-like hierarchy in a hierarchy. And then you actually find that most biological networks tend to be more democratic than autocratic. And as you get more complicated organisms, like you go from E. coli to yeast to worm to human, you actually get an even more democratic structure. And the rationale here is not that these organisms are democratic, it's that this structure is more robust. to kind of knockouts and things like that. And also, one of the things you also see is that there's a tremendous premium on information, easing these information flow bottlenecks in the middle layers and having collaboration between nodes in the middle layers. And what's neat is after we did this, if you zip all the way to slide 45, we were mostly looking at biological systems, but then, for fun, we, opened up some, social science journals and stuff like that. And lo and behold, you can find lots of articles where people talk about, well, if you want an effective organization, you have to have your middle managers collaborating and talking to each other and not have it be, an autocratic thing. And again, I don't know if this is absolutely true in, corporate or social settings, but there is an intuition, that you need to have that middle layer kind of working together and collaborating to get an effective organization. And I think it's neat that you can see it, rigorously in a biological system, and then you get that nice intuition in a social setting.
I'd recommend that people go and make sure that they download and go to slide 45. It's really a bunch of birds sitting on different levels of hierarchy, and then there's a happy bird on top, and then the birds that are underneath are just a little bit white, and then the ones at the bottom are covered with white. It's pretty. That's amazing. So you've taken us through a really important sort of a series of concepts that help us better understand the complexity of life, and I really appreciate it because we're at the verge of going from the single gene, single function element of science, of life sciences, and we need to really get an idea of how to move to the systems biology. And of course, when I was in university, even at the PhD level, systems biology was just starting. I think the government of Canada invested in the first gene chip laboratory to look at how genes were being expressed, sort of on a genome scale. And I think that the human genome hadn't even been completed yet. And now there's mountains and mountains and mountains of data coming in from proteomics projects where they're looking at what proteins are being made in what cell at what time, what disease state, genes are being expressed, or gene chips are collecting data, what genes are turned on, turned off, which cell lines, which ages of the cells, what disease states, what conditions of environmental conditions. I mean, there's so much data, and unless you, I highly recommend that everybody in the field of life sciences that listen to this twice. If you are like me and a reductionist, because to really take advantage of the next round of technology, life technology and biotechnology, we're going to really, if you want to get to that stage where it's like in the movie Blade Runner, where you can manipulate a whole organism, create a synthetic snake, or be it whatever, or tackle a disease that's a complex disease. It really helps to be able to grab that image. And for example, when it clicked to me when I could understand the structure of DNA and how I could visualize it, then it became clear genetic engineering was something that I could do, and then I could expand and do a lot of molecular biology. But to get to systems biology, this is the pathway that we have to take, and get an understanding of how these networks interplay and the importance of the components of those networks. But this could apply to what you found in life, having had billions of years of evolution, you could translate this. FedEx does a pretty decent job, but I don't necessarily think the server farms at the Apple in North Carolina are really optimized. And if you could help them, I bet you could. Really, you've got the algorithms that help you evaluate and calculate bottlenecks. Just that alone is pretty fantastic.
One thing, I don't know if we'll have time, but I'm super keen to show one or two more slides, and that is the next comparison actually is exactly what you're you're alluding to is to make the comparison not only with social situations but also with computational systems, because I don't know. Do we have time for? Yeah, this is kind of cool. So I'm quite proud of this. I think it's a fun thing. So, people often they talk about the genome as the operating system for life. It's the ultimate operating system, and what we've been talking about is how do you understand how the genome works in terms of these networks. And so the next step in kind of this logic would be, well, how can I use the same mindset to understand how a computer operating system works, right? Because now I actually have the computer operating system, and do I see bottlenecks there? Do I see hierarchy in how a computer operating system works? And so what we did is exactly what you're alluding to. We took the same machinery and we went into the Linux kernel. Now, the Linux kernel is very cool because let me tell you a few reasons why it's very cool. First of all, it's completely open source, right? So we can do whatever we want with it. And in particular, there's pieces of software that we can run in it where we can watch the regulatory network in action. We can watch which routine in Linux calls what other routine or regulates it, right? And then how that regulates that. And so there's a nice analogy between the gene regulation of transcription factors regulating one another and subroutines regulating one another in Linux, okay? So we can make that analogy, and then we can see, does Linux operate the same way that, say, E. coli or yeast does? But what's even cooler is now in E. coli or yeast, we can watch them evolve, right? We look at, remember we looked at how in human the parts that vary a lot were on the outside of the network, and we can do that in yeast and E. coli too. In Linux, we can do the same thing because we've watched Linux evolve over the last 20 years. We have every single version of the kernel. So we can take all of the Linux kernels and we can ask for each subroutine how much does it change between each version of the kernel. And so it's just the way we look at a gene evolve in terms of like which genes have more mutations and base changes, we can see which routines in Linux have more changes too. And so it's a very nice, I think, a very nice analogy that you can make between these two things.
So Linux is living, potentially.
Say what?
Linux is alive.
Well, no, it I mean, to some degree, you can see it like that. I mean, I actually, think that it does behave like that. But what's really neat is now, if you go to slide 48, okay, so I showed you the regulatory hierarchy in, Saccharomyces cerevisiae and yeast and E. coli, and it's a pyramid with a few things at the top. And now, remember, E. coli was the same way. There's a pyramid. There's a few things at the top, lots of things at the bottom. And remember, the governmental network was just like that too. There's a few guys at the top, and not that many more people at the bottom. Linux is completely different. The hierarchy in Linux is upside down. It's completely different from the type of hierarchy that we're accustomed to. And it actually, well, in a sense, E. coli is like Linux, but actually you can see how a man-made construction here is completely different. So what does this mean? There are many things at the top that regulate, and there's only a few workers at the bottom that take all the orders, right? Very upside down, okay? So now, what does that mean? So if you go to the next slide, I'll just give you a little bit more intuition. What happens in biological system is you have the big hubs, the things that have a lot of connectivity are regulators. There are things like, for instance, there's a gene in E. coli called CRP. This gene regulates a lot of things. It has a lot of connectivity in the network because it gives a lot of orders. And that's kind of the situation we're most accustomed to in a social situation. The people with the most connectivity tend to give orders out to a lot of other people. But what happens in the computer operating system network is completely opposite. The things with the highest connectivity are the things ordered around the most. And what are those things? Well, if you program a computer, you're probably all used to this. routines like printf or printk, or the routines that you're constantly calling and ordering to do something, these are the routines that, or these are the parts of the operating system that have the most connectivity. It's completely opposite, right? So because what happens in computer networks is or computer operating systems is we develop these reusable components that we really reuse many, many times. And because of that, they get tremendously high connectivity. And we get this inverted pyramidal structure. So it's a very different structure, actually. And it's interesting to see how different it is. And now it gets better. So if you go to the next slide, that's slide 50, one of the things that happens, so what's kind of happening here? In Linux, we're reusing the same routine over and over again, like printk. We're constantly calling it, right? So if you can kind of make these calculations, which I won't go into detail, we can kind of figure out the overlap of all the sub-hierarchies and the amount of node reuse, so the amount of routine reuse. But the bottom line is that in Linux, the routines are used much more than E. coli. So E. coli, you might think of a little like sub-pathway or gene is regulated or is used a lot, but actually it's not participating in many different things. It's actually, the bottom is very fat. Okay, there's not a lot of reuse. Now the next slide, actually let's just zip all the way to slide 52. That slide has the punchline. So what we can do is we can look at each of the we can look at each of the genes in the yeast hierarchy, and we can say how quickly they're evolving, which ones are very conserved. And what we've kind of found is that the genes that tend to evolve more slowly, they're under more constraint, are the ones that have more connections. That was the thing I showed you earlier in the human the human interaction network.
Yep.
In Linux, it's the opposite. It's bizarre. The things that change the most are the things that actually have the highest connectivity. It's bizarre. And
I guess people keep futzing with them.
Well, no, that's exactly the point. So let's go to slide 53. Next slide. That's the point. So what happens in Linux is there's a few routines that are used over and over and over again, and in every version of the kernel, people keep futzing with them, and they keep messing with them. Whereas in E. coli, the things that tend to be used more, people don't touch. And now you might say, why is that? Why is the difference? And it's actually very interesting. There's a reason there's a difference. In E. coli and in yeast, the changes that are made come about from evolution. They come about from random changes that are just random things, right? So, you make a random change, you have no guarantee that change will be good or bad. That's the way natural selection works, random change, right? Whereas in Linux, it's a completely different situation. This is intelligent design, right? Every change that's made is not random. That change is made by a designer. It's made by an intelligent designer who knows that change, or at least intends that change is not going to be deleterious. It's actually going to be beneficial, right? So actually, I know I'm throwing around pretty big words here, but it is the case that in Linux you really do have an intelligent design scenario, whereas in E. coli you have random evolution, natural selection, random evolution, and you really see the difference, I think, in the structures because of these two things. And now the final thing that happens is you might ask yourself, well, one of the neat things about E. coli is that it's robust to all those random changes, and that's the whole point of this. A very small amount of reuse of components. So you can make random changes to the E. coli hierarchy, the organism still lives. It's very robust. Whereas in Linux, if you tried to make random changes, it would fall apart in a second. And even if you make intelligent changes from an intelligent designer and you screw up, you'll mess up the thing. And that's, I think, why complex software systems are actually not that robust. You see?
That's amazing. I'd love to see how other operating systems fit within the scope of and correlate potentially to robustness. Because that would settle it for sure.
We wrote to the guy at Microsoft. We did. We actually talked to him. And they said that they would send us, there's a version of Windows that you can get that's open. And we could do the same thing with Windows. But we haven't gotten that yet. But I'd be very interested to see what Windows is like.
Well, Windows has a lot of legacy stuff. And its function is based, I mean, I guess what's powerful in Windows is its legacy, its ability to run version 1984 of an app. I mean, you could take, I saw a video on YouTube where a person took, they used a virtualization tool, I think it was VMware, and they installed Windows version 1, and then they put on some apps. They installed version 2, version 3, 3.1. Then they went to 95, 98, 2000, and they kept checking back to see if the apps would still run, if it was robust. And there was some versions of, I think, Windows 98 that weren't so robust, and then things got back to XP and things started to work again. It'd be interesting to see if the an approach to legacy code versus starting from scratch and starting over is the way to go. But
it's very interesting. I mean, it's very, very interesting. How we're developing these very complex networks and operating systems now, and people talk all the time about are we building robust things? Are we taking into account to some degree? I don't want to say the lessons of biology, but the ideas of robustness that you get from biology. I think it's quite interesting.
Yeah, it might make you wonder what approach is better, right? To do a synthetic design of an organism from scratch, or to use millions of years of small random changes to build a robust system. And clearly that it's worked. I mean, look, we're alive today after 4.5 billion years, right? And the chemistry is still moving. So it's a pretty strong argument for the survival of the fittest approach to designing the perfect organism, right? Versus, but I don't want to get too metaphysical here. Intelligent design, unless that intelligence is perfect, then somebody that knew that robustness came through random changes, the intelligent designer would say, "I'll let it go random," and to build the perfect.
No, I mean, yes, that's an interesting interpretation. Yes. Yes.
So, wow, your analysis here is absolutely fantastic into the inner workings of life and how these interplay. I never thought we'd get to that point, and that's it. One of these days, I'd like to ask you how you collect or grab the data with respect to a gene and its relative importance. I suppose you go through the databases and look for the number of references and taglines for each one, and sort of create scripts that can go out and fish out that information. But we'll save that for another show, because I'd really like to talk about, if we have another 10 or 15 minutes, about biosecurity, right? And I'm going to suggest that this be the intro to a Security Now episode, and we have to get Eileen and Steve Gibson to connect with this and maybe take it from our 10 or 15 minute introduction to this concept and expand on it, because this really is a matter of security, and I think it would be really nice to introduce it here, and then if you could, cross the Skyposaurus from one screen to another and take it from there, that'd be a lot of fun. So perhaps you could give us some of your thoughts of where security, and molecular genetics interplay.
Yeah, well, I'm as I mentioned to you before, I'm quite passionate about this subject, and I'm very interested in it. recently there's been the advent of this thing called personal genomics, which is essentially the ability to sequence an individual's genome fairly cheaply. And this is an amazing technological revolution that's happened in the last decade, mostly driven by the ability to sequence DNA ever more cheaply. And so now we're in this world where very soon everyone probably will have their genome sequenced as a matter of course in the context of medicine. And you might say to yourself, well, that sounds like a good thing. Maybe people will be able to look at the genome and help with diagnoses and so forth. And I definitely think that's going to be the case. And it's also going to be the case that with a very large amount of genomic data around, there will be tremendous opportunities to mine that information to find very subtle and very interesting correlations that potentially are going to be very informative in terms of people's health. But one issue that's not as obvious is, well, if I have my genome sequenced, or if I have all this genetic information floating around the world, well, what does it mean if other people find out about it, or how does one think about this information? And people haven't really grappled with this issue before. And it turns out, I actually think if you think about it, you're going to realize that this is going to require a wholesale change in people's thoughts about, I think, about privacy and sequencing. Because first of all, genetic information is very fundamental. It's probably the most fundamental information you have. It's more, why is that? Because you can't change it, basically. You can change your thoughts, you can change your clothes, you can do all sorts of things, but you can't change it. And more than the fact that you can't change it, you pass it down to your kids and to your grandkids and so on and so forth. And so, your information actually is beyond you, right? It is associated with your relatives. And, right now, people look at genetic information and they kind of think of it as to some degree, a curiosity. There's people who've, sequence their genomes, or they do what they call SNP chips, which are sort of like cheap sequencing of their genomes. And they, they put the information up, and people look at it, and it's essentially incomprehensible. It's 3 billion letters of AGCTs. It's even more incomprehensible than the parts list I started out talking about. And people don't really know what it does, and they have some ideas and so forth. And right now it's frankly a lot of fun to look at it and to compare it and so forth. But I think what is not appreciated as much is that knowledge is going to, or science is going to go on, and certainly with more and more information piling up, people are going to be able to do more and more sophisticated types of data mining, and this type of data mining will enable them to find all sorts of subtle correlations in your genetic sequence that potentially will predict your health, or just all sorts of your proclivities. What you look like, how you behave, and so forth. And they're probably not going to be able to do it now, but maybe 20 years from now, 30 years from now. But the point is, once the information gets out there in the world, you can't take it back, right? So if I sequence my genome and I put it up on the web, or it becomes something I share, right now it seems like a lot of fun, and it's actually a good thing to do. But 30 years from now, potentially people could mine that type of information to find out all sorts of things about either me or my descendants. And so because of that, I think that people really have to think about how to protect this information. Now, it's actually a very subtle security thing, because on one hand, we want to protect this information. On the other hand, of course, we want to have this information out there for the world so that we can mine it, right, collectively amongst all people, to find very interesting, or not interesting, important correlations that could lead to better medicine, or useful correlations that could potentially lead to all sorts of useful drugs and so forth. And so we want to have a situation where people can do research on genetics, and also find that information interesting, and use it without compromising their privacy. And I think because also the scale of this information, a human genome is 3 billion, or actually 6 billion bases. it's actually hard to think about how to properly protect it, how to move it around, and so forth. And I think this poses a lot of very interesting privacy and security challenges. And I'm quite passionate to, not so much to get the negative word out that this is a problem, but just to get people to think about this and to try to come up with a framework to deal with this information properly before bad things happen, and a lot of genomics gets essentially a bad name, and genomics and data mining. And I think even now, actually, I'm essentially a data miner, and I see more and more that data mining it gets a very bad name. People associate it with pop-up ads or junk mail in there.
No, they gotta flip it, right? Do you want to base your medical decisions on your parents' history, or do you want to base it on your current molecular map, on your parts list, and on your bioinformatic data?
I think that's actually very interesting. That's the famous nature versus nurture debate. To what degree are you determined by your genes? No one knows the exact answers to this, but it's definitely the case that your DNA is going to be associated with a lot of things, and in particular, it's going to be associated with a lot of your physical characteristics. Your sports ability, your musical ability, your susceptibility to certain diseases.
But your DNA is also subject to change. If you take a mutagen, like you smoke a cigarette, right? You're mutagenizing yourself, and you're exposing yourself to changes in perhaps the non-essential outward non-hubs and non-bottlenecks. But God forbid you change a hub or a bottleneck gene, then you're
that's certainly true. You can certainly the environment and mutagens and whatnot. That's certainly something that affects people's health. But I do think that their underlying genetic code is very, very fundamental. And it's interesting to walk around with this type of fundamental information and to think about, well, how should it be used, and how what's the right way of protecting it. And we were talking a bit earlier, and I was sort of saying to people that it's kind of almost like the advent of photography. Before photography, there was no concept of preserving someone's image. And, the concept of like paparazzi or the privacy of your images was never something people would think about, because you just couldn't preserve that at all. Maybe a painter could paint something, but it wasn't really a meaningful thing. Now, people walk around every day with essentially a number of cameras. I actually carry, since I carry an iPhone and things like that, I actually carry at least a camera all the time with me, and people constantly just take images of things, and they don't think anymore of the those things being private or that type of data. And I think we're soon going to be in the same world with sequencing, where we're going to be able to sequence everything, and we have to think about this. And here's some interesting little tidbits just to think about. Imagine you go to a store. We're accustomed now to going to say a supermarket, and there's a camera in the supermarket. Watches, security camera. But imagine the supermarket did something like they said, well, we're going to take the DNA on all the shopping carts, and we're going to sequence it. And we're going to take all the sequenced DNA, and then we're going to do very detailed correlative studies to try to predict which types of people tend to congregate in which aisles with which foods. There's probably a very clear relationship between chemoreceptors and what foods you go in, and maybe they're going to, they're going to try to market certain things related to certain genotypes. It's kind of cool in a way, but it's also kind of scary, okay? And,
it is nuts. It's crazy.
Yeah, that's sort of the issue. I think it's good to have a discussion about this before it actually happens. Or another type of instance would imagine you go to a friend's house and you decide for fun you want to take a swab and just swab along a table in that house, and you take it home and you sequence it, and you get a picture of all the individuals that walked into that house, right? Because they leave a trace, they leave a sequence, right? So you essentially have like a DNA fingerprint of everyone who walked into that room, right? Again, it sounds kind of cool, but what do you think of that? I mean, good or bad? I don't know.
I don't know. I think it's good. I think we need those genomes. We need them quick. I want to base my health condition, my medical strategies on my predicted and planned future, rather than the randomness of not knowing what's going on. I think it will revolutionize healthcare. It will bring healthcare costs down, and it's important to have. And if it helps market to me the things that I need or want, great. But as long as there's a law that says I can't be discriminated against because these are my genes. And then it also helped us, you and I, form a company to make some DNA spray where we take some, hamster DNA, and we'll mix it in with a suntan lotion. And then when we go and use that little trolley at the market, we've confused people.
Oh, yeah. Well, no, but think about this. Think about this one thing, though. They passed GINA, which is the Genetic Information Non-Discrimination Act, and you're probably not going to get discriminated on based on your DNA. But what if your DNA is sequenced, and let's just say, for argument's sake, you're an outgoing person, and you decide you want to put it up on the web, and maybe, I don't know, 30 years from now, a descendant of yours, maybe a granddaughter or whatnot, they do some study and they find that a particular allele or variant of a gene might be associated with some, I don't know, schizophrenia, some mental disease. And then your daughter does a Google, and she realized that, and your daughter's friends realize that, that person has that allele, and they tease them in high school. You know how people are in high school. Discrimination is the way high school works. It's basically how people behave in high school. And it's not something that you can legislate again. It's something that you would want not to happen and want to have prevented from happening.
I guess, and I guess you have to educate, and we will develop a new level of a paradigm for ethics and way of treating other people. We know that certain things are good and certain things are bad and not legislated, and we're sort of become part of our moral fiber. And I guess we'll have to abide by that. Hey, I'm going to recommend that Steve Gibson take this on because there's a lot of really, really important implications here in terms of with respect to security, and it will go well beyond just data.
Just the final thing I'll say, the key thing is I agree with you, but I just want to add that I don't really think that at the end of the day, you can tell people to behave ethically, but I don't think that's actually the solution. I actually think the solution is computer technology. I think that people have to develop a technological system for sharing this type of information and mining this type of information without compromising people's privacy. I don't think you can just simply say, well, educate people and they'll behave responsibly. I think you have to develop computer security set up so that you can deal with this information properly. I actually think my personal view, it's more of a technological issue.
Well, we have to go. Coming up next is TNT, so we have to let it go. We'll have you back on. Well, I'd really like to thank you for coming on because you're clearly a thought leader on in the world of bioinformatics here, and it's a great privilege to interact with you here on the show because I've just learned an enormous amount, and I can see why I can see how valuable it would be to get go through an entire lecture series with you at Yale, and I really appreciate you sharing that time with us here.
I don't think my students would agree with you, though. I think they fall.
I'm sure they would, and then some, because they get more time with you. So thank you very much for coming on.
Okay, thank you very much. I had a good time.
And good luck on Security Now. blow them away.
Sure thing.
In a good way. I'd like to thank Bert McQuinn for handling the audio and video boards today and the recordings. The team that make this possible: Leo Laporte, Lisa Kinzel, Frederic Louis, Aline Rivera, Tony Wang, Mike Taylor, John Slanina, Jeff Stewart, and Jason Howell, and the rest of the team at the cottage and soon to be at the Twit Studios. If you have any comments or suggestions, I can be reached at Marc, M-A-R-C, at twit.tv or at Twitter at Marc Pelletier. I'd like to also thank Phil Pelletier and Will Hall for the opening and closing themes. For Futures in Biotech, I'm Marc Pelletier.